Hello, my name is Hiroki Ota, a professor in University of Tokyo. Today, I will talk about the Jomon genome, the people in the history of East Eurasians. Uh, Before that, I'd like to introduce myself. I got PhD in University of Tokyo in 1997. And then after that, I visited the Max Planck Institute for Evolutionary Anthropology, 1999 to 2001. Then I moved to Yale University of School of Medicine. Then I came back 2005. And then now I became the professor at the University of Tokyo in 2019. This is the building that now I have the laboratory, very old but classical building. It says the science, Department of Science, biology, biochemistry, and so on. This is the laboratory homepage. And then we have two assistant professors, Kae Koganebuchi and Yusuke Watanabe. Our main theme is primary ancient DNA, ancient genome analysis. And then also we examine modern minorities, especially in East Asia and Southeast Asia genome. And then now we are trying to make Jomon-like iPS cells to examine some difference between Jomon people and modern Japanese. So let's start the today's topics. As you may know, Homo sapiens were born in Africa 200,000 years ago. Then Homo sapiens expanded from Africa to Eurasian continent in 50 to 60,000 years ago. Then Homo sapiens spread into western part of Eurasia and the eastern part of Eurasia as well. Then regarding the eastern part of Eurasian continent, we think there were two routes of human migrations. One is southern routes, and then another was southern routes, northern routes and southern routes. Then here, as you see, this is the Himalayan mountains, and then here you can see the Lake Baikal. And then this is the archaeological stuff. Around the Baikal, Lake Baikal, you can see this kind of stone tools, which we call microblade. This is very specific ones that are found around Lake Baikal, which around the late Paleolithic period. Then that's why we think the human took root of northern part as well as southern routes. Then, interestingly, microblade has been found in Japanese archipelago. Well, a kind of microblade has been found in Japanese archipelago. So the people who used the microblade might have reached the Japanese archipelago. So we can imagine that the people in this area with a question mark are the hybrid between the people who came from southern route and the northern route. But here is a puzzle. So now we have a huge data set of modern human genome. Then genome data from modern humans only show the southern route. For example, the Hugo Pan-Asian SNP Consortium published this paper in 2009. Then in this phylogenetic tree showed only Southeast Asian origin of East Asian. As you see here, green colored ones are Indonesian and the Philippines and Taiwan, and that means those are kind of the Southeast Asian. Then red colored one diverged from these that represent Thailand people and the southern China, and then also blue colored one separated from these, which are Thai people. Then this sky blue colored ones, China and Thailand, and then purple colored ones are China and Taiwan Aborigines. Then finally branched out Japanese and Koreans. So that means the East Asian were originally born from, branched out from Southeast Asians. Thus, genome data, modern human genome data show that only southern roots origin of East Asian. So then we focus on the Jomon people to test, to look at the East Asian people in history, because Jomon people were, Jomon people are thought to be a descendant of the late Paleolithic people who lived in the Japanese archipelago since around 38,000 years ago. So then the Jomon culture started around 16,000 years ago. Then they were fishers and then hunters and gatherers. This timing is around the final stage of last glacial period, and Japanese archipelago separated from the East Eurasian continent around that time. So Then so that's why the German people were isolated since 16,000 years ago, then up to 3,000 years ago, because after 3,000 years ago, migrants with rice cultivation came into Japanese archipelago from the East Eurasian continent, probably through the Korean Peninsula. So therefore, the bone of German people might have maintained the oldest DNA of East Eurasian people. So we challenged to extract DNA from a bone of German woman excavated from Ikawazu Shell Mound site. We named this woman as IK002. This woman had a baby born on the breast, on her breast. The baby, we named the baby as IK001. So this is this woman was called IK002. Well, genome analysis was done in the clean room exclusively provided for ancient DNA samples. To remove modern DNA, modern human DNA, as you see, the clean room was has UV irradiation before and after the experience, because the contamination, modern human contamination, is most risky problem for ancient DNA analysis. This is the flow chart of ancient DNA analysis. In the clean room, we extracted the DNA, and then the quality check by, we did the quality check by bioanalyzer, and then we prepared NGS library in there. Then the, mainly we extracted DNA from Petrus bone around here. Then the did shotgun sequencing by MiSeq, and then after that, if you have, we have the more than 1% of indigenous DNA, then we move on deep sequencing of by HiSeq and NovaSeq. Then the data analysis were done by Martin Schikola in Copenhagen University, and then Takashi Gakuhari in Kanagawa University, and then Shigeki Nakagome in University of Dublin as a collaboration. Then we finally obtained the draft sequence, draft genome sequence of IK002, and then firstly reported the sequence data in Science 2080. The main topics of this paper was the prehistoric peopling of Southeast Asia. So the German genome sequence data was published together with 25 ancient genome data from Southeast Asia. this is the figure that includes the data we supplied. This graph shows the ranking who is close to the 8,000 years old Hoabinhian hunter-gatherers excavated from the archaeological site in Laos. Hoabinhian is the name of the culture which was in Southeast Asia. That is what Hoabinhian culture was hunter and gatherers culture. The closest one was group 3, which is a group including ancient DNA from Southeast Asian bones. Then group 3.1 and group 4.1 are the second and the third closest ones. Both of those are ancient DNA from Southeast Asia. That's not surprising, but surprisingly, Jomon individual, the Jomon individual came into the fourth one. Those were modern humans, mainly from Southeast Asia and East Asia. So the data show a very close relationship between ancient Southeast hunter-gatherers and the Jomon people. So considering this result of the former paper, we reanalyzed the genome data of IK002 and published, reanalyzed the data, then published this paper as maincast. IK002 was a maincast of this paper. So let's go back to the figure. I explained the puzzle once again. The primary question of this paper was whether IK002 was a descendant of southern root or northern root people. There should be two routes, as well as I mentioned, into East Eurasian based on archaeological evidence, but the genome data from modern humans show only the southern route. Therefore, we examine the Jomon genome because the Jomon people was isolated for a long time in the Japanese archipelago and could have the very old East Eurasian genome. So to test it, we used two other ancient genome as proxy. One is Malta individual who is 23,000 years old. And then another one is the Hoabinhian hunter-gatherers excavated from Laos, Southeast Asia, which were 8,000 years old. Namely, we used two ancient genomes as a proxy of southern and northern and southern roots, as you see. So in the phylogenetic tree reconstruction, if IK002 branched out from Hoabinhian branch, then IK002 is involved in southern root lineage. Meanwhile, if IK002 branches out of Malta branch, then IK002 is involved in northern root lineage. So then we reconstructed the phylogenetic tree by maximum likelihood method, like this. Then I would like to explain the data. Here, Mubati, which is African people, and then this is outgroup of this paper, of this tree. Then first, Ust-Ishimu separated, diverged, which is Upper Paleolithic ancient bone from northwest Eurasian continent. Then this branch had two branches like this. Then first branch include Malta, which is a proxy of northern root. This is a northern root. Then these are cluster of southern root. In southern root, first of the southern root, the first, the Onge and the Hoabinhian hunter-gatherer diverged. Onge is a modern hunter-gatherer in Southeast Asia. So then after that, Tianyuan, which is Upper Paleolithic individuals from China around Beijing, came here, which is 40,000 years old, around 40,000 years old. Then after that, Kusunda diverged. Kusunda is a minority in Nepal. Then next, Jomon diverged. Then after that, Chokpani and Sherpa diverged like this. Chokpani is a ancient Nepal people individual, and then Sherpa is a minority in Tibet. Then Ami, which is Taiwan Aborigine, and the Han Chinese came here. Then Japanese separated like this. This arrow, yellow arrow, represent gene flow from Jomon. So that's very reasonable result because the Jomon is an ancient population of in Japanese archipelago. Then those are all those were then Devil's Gate Cave, which is the ancient bone from Northeast Asia, East Siberian ancient bones. Then these are Native American, well, maybe ancient ancestor of Native Americans, which were found in American continent, North American continent. So, and then for these people, gene flow came Malta, like this, which is the already reported in previous studies. So this represents like this. Thus, the phylogenetic tree showed IK002 is involved in southern root lineage. That means IK002 is a descendant of the people who reached the East Eurasian continent through the southern route. So the second question is whether there was a gene flow from Malta to IK002. Previous studies showed that Northeast Asia and South Native Americans had substantial gene flow from Malta, as I mentioned. So if IK002 had the Malta elements too, then it is an evidence of ancient hybridization in East Asia between southern and northern lineage. So to test, to investigate it, we used a couple of statistical tests. These statistics is one of them, which has used to examine Neanderthal hybridization to Homo sapiens. The formula is here. If you are interested in, please look at the original paper, but I explained briefly the principle. For example, if in the site in the genome, chimpanzees have G allele, and the Neanderthal have G, and then African modern human have A, and then non-African humans have G or A. Then what we assume what happened. First one is that G to A substitution occurred after divergence between Neanderthal and modern human here. But in modern human, especially in non-African modern human, if you have both G and A allele, then you might think A to G substitution occurred. This is which we call back mutation. But based on the mutation rate in human, it's not plausible. Then what we say? We can say gene flow from Neanderthal to non-African people after out of Africa migration. So we count this kind of sites in genome, then calculate these statistics like this. So this is the example of Neanderthal and human hybridization, but we examined this kind of stuff in East Asians and Southeast Asians and the Northeast Asians, like this. This is the data. Red colored ones are the values representing gene flow from Malta into population X. X are those populations, as you see. Blue colored ones, the value representing gene flow from population X to Malta. Then here, this axis is zero. If you have a significant minus value, then it represents substantial gene flow. Then, as you can see, almost all Northeast Siberians significantly had minus value of red-colored ones, which agree with previous studies. Almost all Northeast Siberians had substantial gene flow from Malta, who is a proxy of the northern root lineage. Meanwhile, no Southeast Asians and the East Asians had significant minus values, suggesting very little gene flow from Malta. Okay, at the bottom, you can see IK002. IK002 also has no significant gene flow from Malta Paleolithic man. So we got the answer to the second question. We didn't have any evidence of gene flow from Malta into IK002. So finally, here, I would like to summarize the results. The two lineage here, you can see two lineage, southern root and the northern root. The root of northern root, southern root, you can see Tianyuan individual from Beijing. Then these are modern populations. Onge, which is Southeast Asians, Southeast Asians, and then East Asians, and then Northeast Asians, which is East Siberians, and then the Native Americans. All are descendant, the people migrated from through the Southeast Asia, as you see. Then the ancestor of IK002 diverged just after the divergence between Southeast Asia and others. So that means IK002 were very old lineage in East Eurasian human populations. The hybridization between northern people and were observed here, that just after the divergence between East Asian and Southeast Asians and the Native Americans, ancestors of Native Americans here. So therefore, we do not detect the evidence of gene flow from Malta in modern East Asians and even in IK002. This is the scenario that we have now. However, we examined only one Jomon individual in this study, and only one proxy of northern root lineage, Malta, only one Malta. So after we published the paper I introduced here, several papers, including Jomon, other Jomon individuals were published. So by reanalyzing the data, we might have the other answer. Further investigation must be required to know the real people in history of East Eurasians. So there is a co-authors, co-workers of Jomon Gino. Thank you so much for your kind attention. Bye-bye.